Thanks so much for the kind invitation and for the introduction. So, as Nancy said, my task today is to talk about our sense of location. It's not a real sense. It's actually not a sense because it's internal in the brain. But nonetheless, the focus of my talk is on how we know where we are and how we find our way from one place to another. And looking into the space, to the brain's navigation system, as we call it, we believe can help us understand not only navigation, but also brain computation in general. So here is our discovery from 2005, for which we received the Nobel Prize in 2014. So the prize was given for grid cells, which we found in a brain area called entorhinal cortex. So these cells are active only when animals or people are in certain positions. What you see in the video is a rat that is running around in a box. It's seen from above. The box is about 1 by 1 meter. And each time a particular cell in the entorhinal cortex is active, you see a white dot appearing on the screen. So you can see that this particular cell is active only at certain locations. They've done clusters of locations, and those clusters have a very, very peculiar pattern. So you can see the pattern to the right. There, black is where the cell is active, and gray is where the rat has been walking around. And you can see the repeating triangular pattern all over the environment, or hexagonal or rhomboidal, whichever you prefer. But it's a periodic pattern that unfolds over the entire space, and in many ways, as Nancy said, serves as a coordinate system for our location in space. So this was 2 decades ago. Today, we try not only to record from one cell at a time, not only to look at the elements of spatial coding. Today, we have technology that allows us to look at the combination of activity among thousands of cells at the same time, for which we use silicon probes that allow us to record from many thousands of places in the brain at a distance of approximately 20 micrometers. And that allows us then to get the activity of, in this case, it only shows 149 cells. Each row is one cell, and black is active, white is not active, and this is over a period of a few seconds. And then you can look at the combined activity of those cells, and in red I've shown one bin of time, and then we can ask what is the combination instead of what is one single cell doing. And it is then possible to investigate those combinations and ask how are they different in different locations, and you can actually from that then infer the activity of the brain or the entorhinal cortex in the internal map. So we'll do that, make such an inference over time. So now you see again grid cells, but it's very, very much slowed down, and you can see that the inferred activity actually has a pattern in time where the activity goes left, right, all the time, where each sweep, as we call it, has a duration of approximately 50 milliseconds. So grid cells are not only static, they actually move forward in time in a strictly alternating pattern that has the advantage that if you do it in this left-right manner, much like bats do with echolocation systems, then it's actually an optimal way to survey the environment and to look forward, not only where you are, but actually also where you're going. But then we ask, now I'm over to unpublished work, is this different when there's something interesting out in the world? So here we have a bait that's hanging on the fishing rod in front of the animal, and you can see the rat that now these sweeps actually target the bait pretty much all the time. So it's much more straight towards the bait, and the left-right alternation is still there, but is much, much more sharply focused. So then we can ask, again, my last data slide, what happens when the bait suddenly goes away? So here you see that sweeps actually target the bait even when the rat lost it for a little while. So you'll see now, rat loses the bait, bait goes back, and the rat gets confused for a little while. Then the sweeps target the bait before the animal actually turns itself. So this sweeping system is not only a mechanism for serving the environment, it actually is also a spatial attention mechanism that allows the animal to focus in space as it explores the environment. So with this, we get from representation of current location to actually navigating the upcoming environment and finding the parts of the environment that are interesting at the moment. So a spatial attention mechanism that is embedded in the activity of thousands, hundreds of grid cells. So with that, 2 seconds left. Thank you for your attention there.
Thank you, Eva, for setting such a good example for the other laureates. So, may I now invite the second speaker, Professor Maybritt Moser, winner of the 2014 Nobel Prize in Physiology of Medicine. Please, Maybritt. Thank
you. Good morning. I don't see you, but... So thank you for this invitation. Thank you to Nancy for this beautiful introduction, and thank you to Edvard for presenting part of what is interesting for my own talk. So you heard Edvard speaking about this grid cell system, the spatial navigation system, and we are both psychologists, and as a psychologist we were trained to think, are these functions, are they inherited or acquired? And that is the question. And if you wonder who these beautiful babies are, they are Edvard's and my grandchildren. So now you recognize these raster plots where we have the spikes of the grid cells. And what is quite interesting is that this, when we look into each time period, then of course each cell varies. So if you record them from one module, that means one size of the grid cell, one map, then you see that the variation within each time period here with 149 cells could be plotted in 149 dimensions. And even 4 dimensions are quite difficult for us to understand. So we need to use mathematical methods to reduce the dimensionality. And when we do that, since this is a periodic system, as Edvard told you about, then what we found, this is just with the UMAP, we have also done topology measures, we found a torus. So the activity has to follow this manifold. And why is this exciting? No, because Edvard said that when you see grid cells, you have to let the animal walk around so that you see this pattern, the grid cell pattern being expressed. A baby rat doesn't move much. But the nice thing is that this torus is active also when animals are sleeping. So that means now we have a tool to ask our question, do we see this internal map in a baby rat that hasn't moved yet? And just about the development of animals, the first sense that they develop is when they're just born, P0 to P2, that's the olfaction. And the ears and the eyes don't open before P12, P14. So we decided we go and hunt for an internal map at torus in baby rats at P8. And this fantastic postdoc, Matteo, he was able to do these surgeries when the animals were sleeping. And in order to find the grid cells, or we don't know, but the internal map, we had to use another mathematical method here, where we asked if you have the cross correlations between the cells, and then you plot in a matrix how these cross-correlated cells are correlated. It's a quite complicated method, but it works. And when we go into these very correlated parts of the matrix, at P10, we found the torus, and not before. And to the right, you see that it's possible to plot the activity of the different cells on the torus and map it out. So we think that this is evidence for Kant. So I would have wished that I could meet him and say, yes, you were right. Because he suggested, comparing to people who suggested we are born like tabula rasa, we have to acquire all senses. He suggested in order for us to experience the world, then we need some faculties of mind to be there from beforehand. And one is about time, and the second is about space. So, do you think he would be happy? I think so. Thank you.
Thank you so much, May-Britt. So may I now invite the next speaker, Professor Erwin Nier, winner of the 1991 Nobel Prize in Physiology or Medicine. Please, sir.
Hello, good morning. I think the two preceding lectures have clearly shown that the brain is a very dynamic system with changes in connectivity occurring on, say, many time scales as information processing is going on. So what I would like to point out today are a few details about this dynamic connectivity on a cellular level. And what it's more or less about is what's called synaptic plasticity. Synaptic plasticity is a term that neuroscientists use to describe the observation that connections of cells between cells are dynamic, depend on the information flow through things. So neuroscientists are convinced that these changes underlie the complex information processing, and this plasticity occurs on many time scales. So research on plasticity became very important and very popular after the seminal discovery of Teddy Lomo and Tim Blyth, that you can induce such changes which then persist over some time by just stimulating synapses in the brain, in the hippocampus, and that by applying a strong stimulus has an after effect, an increase in synaptic transfer over many minutes up to hours. So following this discovery, there was a long debate about what's changing. Is it changes in the postsynapse, changes in sensitivity of the postsynaptic cell, or is it changes in the release of neurotransmitter? And the attention focused for many years on the postsynaptic side, namely the changes in glutamate receptor density and opening probability of ion channels on the postsynaptic membrane in response to the release of a neurotransmitter. What I want to point out is that although there has been described many mechanisms of plasticity, both pre- and postsynaptic, the attention was very long focused on the postsynaptic thing. But in my lab, we tried to do the opposite, to look at the presynaptic side and to put this on a new basis. What happened is that over the last few years, there have been many pieces of evidence from electron microscopy, from molecular biology, from physiology, that the process of release is actually quite complicated, that when a vesicle is supposed to be released by an action potential, it first has to dock at the right position. It has to develop molecular machinery which then, once it is already built up, allows a vesicle to be released upon an action potential. So, in particular, what has been shown is that this process is dynamic, switching between several stages of maturation. And my main point is that in the past, the methods to study this presynaptic mechanism of docking, priming, and release was viewed too simplistic. Many studies did not allow to mirror the dynamic properties of the docking and priming process. In particular, what's shown on the right side is that at the core of these considerations is the protein MUNC13, which is more and more clear that it is an important mediator of the docking process, and in particular that it is regulated in many ways by having binding sites for second messengers such as calcium, diacylglycerol, and PIP2 phosphoinositides. And so that's the most important point, I think. And in this priming-centered view of neuromodulation, we showed that both long and short-term plasticity can be regulated, can be influenced by this regulation about second messengers. And let me just remind you, this is a slide from Matthijs Verhagen's laboratory, which just points out the multiplicity of signal pathways through G proteins and agonists leading to the second messengers. And of course, there is influx of calcium is an important element, but there is also this protein MUNC13, which, as I pointed out, is regulated in many ways. Thank you for your attention.
Thank you, Owen. So now let's welcome our next speaker, Professor Adam Patapochian, the winner of the 2021 Nobel Prize in Physiology or Medicine.
Good morning. So I'm here to tell you that your sense of touch is amazing. You have the ability to sense indentations at your fingertips that are 1/500th the diameter of a human hair. On top of this, we rely on sense of touch for our emotional health. And this session is called the Six Senses. One of the reasons is that this, my favorite sense, proprioception, which is a sense that most of us don't even know that we have it, is very important. And specifically, this is how you sense where your limbs are in space. So I can, for example, close my eyes and touch my nose. If you think about how I did that, just from stretch sensors in your muscle, you have this image of where your limbs are and what space you occupy. These are all mechanosensory processes that in the lab we were very interested in how you actually sense this at the molecular level. So how do you sense a gentle breeze or a cactus pricking your finger? And we know that there are these sensory neurons that are both sides of your spinal cord, they're cell bodies, but they send processes all over the body, gathering information from the periphery and sending it to the spinal cord and the brain. And at the tips of these neurons, there are these specialized proteins called ion channels, very fast signaling molecules that can be activated, in this case, by physics, either temperature or pressure. And this is what my lab has been interested in. David Julius's lab and my lab first identified temperature-activated ion channels. In 2010, we found the first mechanosensors that we called piezo-1. So there's two of these ion channels called piezo-1 and 2. They are, I think, fantastic molecules. They're very large. They're about 2,500 amino acids, and they traverse the membrane 38 times each, so they have 38 transmembrane domains, more than any other protein in your genome. On top of that, 3 of them come together to form one of these molecules, so this giant sitting in the plasma membrane. And the place where all 3 protomers touch each other is in the C-terminus, the center where the ion conduction happens. The rest of it, you see these ribbons are these transmembrane domains that are extended into the plasma membrane, kind of like antennas for membrane tension. As you will notice, very interesting feature of the channel is that they have this puckered orientation, and that's thought to be key to these proteins being professional mechanosensors. So they bend the membranes, and then when they experience stretch, they actually flatten to activate the ion channel. The remarkable molecular tool that evolution has come up with. And various groups, including Rod McKinnon, Bai-Long Zhao, and our group in cells have shown that this remarkable flattening actually happens in response to tension. Now, if you love a protein as much as I do on this guy, you gotta brand it on your own body. And I wanna show you my tattoo of piezo. And you know, I do this just because when I have power slides like this, I can show it. But what if I'm in a restaurant or a bar or a dinner party and I want to demonstrate how this ion channel is gated? So this is the closed conformation in the bent configuration, and any stretching will flatten the ion channel and ions can come through. So I encourage you all to brand yourself with your favorite protein. So after identifying this ion channel, we wanted to know what it does, of course, and we've done this both in animal models as well as from clinical studies. I have very little time, of course. I could tell you in mouse models, for example, deleting piezo-2 completely gets rid of the ability of these animals to sense touch and proprioception. How about in humans? My colleagues, Karsten Bonneman and Alex Chesler at NIH, identified mutations in piezo-2. So these are null mutations, no functional protein, where just like we'd shown in the mouse, they have complete lack of touch sensation and proprioception. And this video shows one of these piezo-deficient individuals showing the lack of proprioception. So she's harnessed for her own protection, blindfolded, and asked to walk in a straight line, something you and I can very easily do, but as you're seeing, she really struggles doing this task. So over the years, we found many important roles for these ion channels that's all related to pressure sensing. This involves, as I said, touch sensation, proprioception. We found that it's required for a specific form of clinical pain called tactile allodynia, when we're currently working on finding small molecules that could be beneficial for treatment of pain. Mechanical itch. We've also found with collaborators many interesting roles in internal organ sensing. Some of them you're not even conscious. For example, you monitor your breathing rate and control it based on this sensing of lung stretch, similar to sensing blood pressure in your system. Some of these other interoception, internal sense organs, you're very aware of. You, for example, are aware of how much your bladder stretch. That's when you go up and go to the restroom. And also digestion. Just to give you an example, these piezo2 deficient individuals do not have this bladder stretch sensing, and so they've learned to go to the restroom 3-4 times a day to avoid accidents. And this is just the beginning. We have so many other projects now studying the role of pressure sensing in our bodies. One of my trainees, Kara Marshall, studying, for example, what's the consequent stomach stretch in how much you eat, when you stop eating. So most of the research on satiety is based on chemical sensing, but these molecules are allowing us to ask what is the contribution of pressure sensing to these physiological processes. Once evolution finds a molecule that works well, it uses it again and again. And we've found, for example, that even plants use mechanosensation. For example, their roots need to decide whether to penetrate the hard surface or go around it. So piezo in Arabidopsis is expressed at the root tips, and without it, this ability to penetrate hard surfaces is compromised. Evolution is indeed amazing. This is a recent reunion from the lab, so all my trainees from the last 25 years as well as current came together to celebrate science. Thank you for your attention.
So with Professor William Kaelin, Jr., the winner of the 2019 Nobel Prize in Physiology of Medicine. Please.
Thank you very much, although I think I have tattoo envy now, so so these are some kindreds of families that have von Hippel-Lindau disease, and each of the filled circles and squares is an individual who's developed any of a number of cancers, typically kidney cancer, blood vessel tumors of the brain, eye, and spinal cord called hemangioblastomas, or tumors of the peripheral nervous system called paragangliomas. Now this disease affects about 1 in 35,000 people around the world, and although it was first described about 100 years ago, it's clear some of these families have been affected for centuries. Now a word about the genetics of von Hippel-Lindau disease. I think most of you know that for most genes we have 2 copies, one from mom and one from dad, and they're organized in 23 pairs of chromosomes. So here schematically are the 2 chromosomes that harbor the VHL gene. People with von Hippel-Lindau disease have inherited a defective version of the VHL gene from mom or dad. In this schematic, it's the maternal copy. But initially, they're okay because they have the remaining good copy from the other parent. But unfortunately, if they're born with one defective copy, they have about a 90% chance that a susceptible cell in their body, such as in their brain, eye, or kidney, will spontaneously mutate or lose the remaining normal copy, and that's the cell that will go on to form a tumor. And as you would predict from the knowledge that inheriting a defective VHL gene predisposes you to, for example, kidney cancer, if you now look at garden variety non-hereditary kidney cancer, you again see that both copies of the VHL gene are frequently mutated or defective, but here both mutational events occurred after conception, in contrast to VHL disease where the first mutation was inherited from mom or dad. And I should point out that kidney cancer is one of the 10 most common cancers in the developed world. Now, I knew from my clinical training, I was a clinical doctor before I became a scientist, that the tumors seen in VHL disease are notoriously rich in blood vessels, and that's because they frequently produce massive amounts of a hormone called vascular endothelial growth factor. And another curiosity is that these tumors sometimes cause patients to make too many red blood cells, so-called erythrocytosis or polycythemia. And that's because these tumors sometimes produce the hormone erythropoietin, which naturally stimulates red blood cell production. And what VEGF and erythropoietin have in common is that they're normally upregulated when cells or tissues are not getting enough oxygen, presumably to try to restore oxygen delivery by increasing vascular density and increasing the oxygen capacity, carrying capacity of the blood. Now, I was told this was a very mixed audience, and no one ever got mad at me because I made them feel smart. So just to introduce a little jargon, so really most genes have the instructions for making a protein, and when your cells want more copies of that protein, they will activate that gene. Technically, the gene is now transcribed or expressed, and that's because a protein such as a transcription factor comes along, binds to the gene, and stimulates the process called transcription, where multiple copies of that gene are made in the form of messenger RNA, and the messenger RNA then drives the production of the protein. So on the left, we have a gene that's being actively transcribed, and on the right, that gene is inactive. Now, I mentioned transcription factors because many of these genes that are normally upregulated by low oxygen, such as vascular endothelial growth factor and erythropoietin I just mentioned, are under the control of a transcription factor called hypoxia-inducible factor, or HIF for short. And hypoxia is just a fancy way of saying low oxygen. And we knew from the work of multiple laboratories as well, including the work of my fellow laureates, Greg Semenza and Peter Ratcliffe, that HIF actually contains two subunits, an alpha subunit that's normally degraded when oxygen is plentiful, hence hypoxia-inducible factor, and a constitutively stable beta subunit that also goes by the name of ARNT. And so based on this and other clues, we and others went on to show that the VHL protein is part of a so-called ubiquitin ligase complex that in the presence of oxygen binds directly to the HIF-alpha subunits, attaches a little so-called polyubiquitin tail covalently, which then serves as a signal for HIF-alpha to be destroyed by a cellular garbage disposal called the proteasome. Whereas when oxygen levels are low or the VHL protein has been crippled, such as in kidney cancer, now HIF-alpha can accumulate, dimerize with its partner protein HIF-beta, and turn on various genes such as vascular endothelial growth factor or erythropoietin. So that was very satisfying, but it still begged the question, how do cells know, if you will, whether oxygen is or is not available, and hence whether or not it's appropriate to destroy HIF-alpha? And in work that we did and Sir Peter Ratcliffe did working in parallel, we showed that when oxygen was present, one of two prolyl residues within HIF-alpha gets hydroxylated, and that then generates a high-affinity binding site for the VHL protein. And at the time, this modification had never been seen as an intracellular signal. We and others wanted to show that the enzymes that do the work here are the so-called EGLN prolyl hydroxylases, alternatively called the PHD prolyl hydroxylases. They split the ambient oxygen and use one of the oxygen atoms to hydroxylate HIF-alpha. Parenthetically, these enzymes also couple oxygen sensing to metabolism because they have an obligation for 2-oxyglutarate as one of their cofactors. So where has this led us? I should point out this system is conserved throughout metazoan evolution, so the same oxygen sensing system is used by flies, worms, you name it. Importantly, it became very clear once we knew the rules that this system could be pharmacologically manipulated. So there are now a number of HIF stabilizers that are orally available that interfere with the prolyl hydroxylation reaction. They've been approved for the treatment of anemia around the world, and they look quite promising for the treatment of heart attack and stroke, at least in preclinical models. Conversely, we now have first-generation direct inhibitors of HIF, and in particular HIF-2, which have been approved for the treatment of kidney cancer, as well as for the treatment of VHL disease. Thank you.